Good day, everyone. Today I'm going to discuss the genetic basis and biotechnological manipulation of sexual dimorphism and sex determination in fish. So, aquaculture has made an enormous contribution to the world food production, especially to the sustainable supply of animal proteins. The utility of diverse reproduction strategies in fish, such as the exploiting use of unisexual gynogenesis, has created a typical case of fish genetic breeding. A number of fish species show substantial sexual dimorphism that is closely linked to multiple economic traits, including growth rate and body size, and the efficient development of sex-linked genetic markers and sex-controlled biotechnologies has provided significant approaches to increase the production and value for commercial purposes. Along with the rapid development of genomics and molecular genetic techniques, the genetic basis of sexual dimorphism has been gradually deciphered. A great progress has been made in the mechanism of fish sex determination and identification of sex determining genes. So we have here the reproduction strategies, sexual dimorphism, sex determination, sex determining gene, sex chromosomes, and sex controlled breeding. So sa mahabang panahon, napaka-importante na magkaroon ng sex kasi isa tong basihan sa mga sciences sa mga napag-aralan dahil most vertebrates nga daw ay gonochoristic and show significant sexual differences in morphology and physiology sa kanilang mga katangian at mga itsura. So, sabi nga rito, sex is the queen of problems in evolutionary biology. And number one, we have the diversity of fish reproduction strategies and their application in genetic breeding. Diversity of reproduction strategies in fish. So, alam naman natin na ang fish is the most abundant and successful group of vertebrates sa buong mundo. Meron siyang 32,700 documented species. So, napakarami talaga. Base doon sa may data na nanggaling sa fishbase.org. So, ang fish din ay unisexual, hermaphrodites, and gonochorist. And majority sa mga isda ay gonochorism and hermaphroditic phenomena. Kasama nga rito yung mga na-reported na species kagaya lamang ng black sea bream or which is the akan to pagrus schedule and grouper Epinephelus coides. Meron din dito yung gonochoristic rainbow throat na napag-alaman na sex steroid level change could reduce hermaphrodites, and the hermaphrodites can produce YY males na by self-fertilization. So, nag-fertilize siya sa sarili niya na nag-generate which is generated from sperm and egg. So we have here the discovery of multiple modes of unisexual gynogenesis and sexual reproduction and the genetic breeding application in polyploid gibel carp. Sabi rito, the Amazon molly or the Poecilia formosa was found by American ichthyologist Hobbs et al. In 1932. So unisexual, all-female population has been reported in about 30 fish species. And these unisexual fishes had been demonstrated to reproduce by gynogenesis, hybrid genesis, or parthenogenesis. Polyploid gibel carp, Carassius auratus gibello, because of the existence of minor but significant portion of males in natural populations, and the discovery of multiple modes of unisexual gynogenesis and sexual reproduction in some gynogenetic clones has been noted extensively. So, in addition, a novel nucleocytoplasmic hybrid clone A+ has been created by sexual mating between clone D female and clone A male, and rapidly multiplied up to several hundred of millions by subsequent more than 5 generations of unisexual gynogenesis. So, imagine that, ganun yung ginagawa nila para lang ma-produce into or mag-multiply siya ng millions. Owing to the significant growth, superiority, and better resistance to mycosporians pathogens parasitized in the liver, the novel clone variety as nominated CAS3 has been cultured throughout China. So, number 2, the sexual dimorphism and its genetic basis in fish. The sexual dimorphism is commonly defined as the difference between male and female individuals. So, doon mo malalaman yung pagkakaiba ng lalaki sa babae, ano may mga differences nila when it comes to their physiology and yung kanilang mga katangian. Sexual dimorphism in fish not only includes size dimorphism, shape dimorphism, and color dimorphism. Siyempre, meron din dito yung kanilang sexual differences in physiology and behavior sa mga kilos nila and sa mga galaw. O yung kilos or galaw nila o yung mga katangian nila sa kung anong meron sila nun. Long-term field surveys and aquaculture practices have observed significant size dimorphism between male and female individuals in more than 20 fish species. So mamaya mapapakita diyan yung mga kung ano nga ba yung mga species na yun. The females mature later than the males so that the nutrient substance in female is able to transfer into body composition and body weight during maturation and reach a larger size than males. So mas mabilis daw mag-mature yung female. And then during maturation and reach a larger size than male. So mas malaki siyang lumaki. And kung mapapansin nga daw, yung kanilang body is talagang mabilis mag-mature, mabilis mag-transform. Males mature later and grow faster in more than 10 fish species. So, mas mabilis namang lumaki yung mga males kumpara sa mga babaeng isda. In some sex-changing fishes, sabi rito, in some sex-changing fishes from females to males, mas mabilis lumaki yung lalaki kaysa sa babaeng isda dahil nga ang malaking male na isda ay naniniwala sila na to monopolize the right to mate with many females. So mas siguro gusto nilang nagme-mate sa more than 1 or 2 females. Ito na yung mga known species with sexual dimorphism base sa kanilang size. We have the size dimorphisms, bigger in males and bigger in females. Sa bigger in males, itong mga species na to kagaya lang ng yellow catfish, XX, yung kanilang sex chromosomes which is XX and XY. The Nile tilapia, XX and XY. So, parehas lang din sila ng kanilang sex chromosome systems. And then the Ussuri catfish, Channel catfish, snakehead, bluegill sunfish, blackfin sandperch. Itong mga ito, kagaya na ng blackfin sandperch, bluefin tuna, dark sliver, and rusty parrotfish, hindi pa na-identify or unidentified pa yung kanilang sex chromosome systems. Dito naman sa bigger in females, andyan yung mga common carp, bustard halibut, southern flounder, rainbow trout, and so on. And meron din dito na hindi pa identified ang kanilang sex chromosomes which are the European eels, spotted scot, and silver barb. And the genetic basis of sexual dimorphism in fish. Sexual dimorphism commonly exists through animal kingdom. So, alam naman natin yan na doon sila nag-exist. Common na yon. And the size dimorphism in vertebrates mainly results from a different growth rate between male and female individuals, and the growth rate is regulated by growth hormone, insulin-like growth factor, and other related factors secreted by the hypothalamic pituitary gonad axis and other tissues. So, sa iba namang mga fish species, sexual dimorphism in color, body shape, physiology, and behavior is also controlled by some key genes. So, nakokontrol sila sa mga key genes ng ibang species. For example, in Lake Malawi, cichlid fishes, the color dimorphism between males and females is probably controlled by the expressional and functional differences of PAX7 gene. And the neo-sex chromosomes in stickleback has significant correlation with the physiology and reproductive behavior. So, sa blue tilapia naman, the hybrid offspring nila is nakapag-produce sila ng more than 93% males na na-applied nila sa kanilang malakihang aquaculture, which is na kinulture sa China. In bass naman, meron silang 100% female offspring na nag-generate by a specific hybridization between different species. And then the artificial gynogenesis and all-female population production. Artificial gynogenesis generally refers to the haploid egg activation by genetically inactivated sperm and activated egg development into diploid offspring after chromosomes doubling. Yung XX or the all-female populations will be produced. The parents have XY and XXX determination system. Whereas yung ZZ naman na lalaki and WW na babae na hindi lahat ay female offspring will be theoretically nagproproduce from parents with ZZ and ZW sex determination system. So kung sa pangkalahatan, ang gynogenetic offspring is composed of all females. So lahat ay puro babae na species. And the identification of sex-specific markers and sex chromosome-specific markers. The degree of sex chromosome differentiation is very low. So, napakababa niya and mahirap siyang i-discriminate through sex chromosomes morphology. So, mabilis din siyang ma-influence ng environmental factors kagaya na lamang ng sa kanilang temperatura sa kanilang tinitirahan o environment. Sex-specific or sex chromosome-specific markers by multiple techniques including AFLP or the amplified fragment length polymorphism, the SNP which is the single nucleotide polymorphism, the RAPD random amplified polymorphic DNA, and SSR simple sequence repeats, and QTL the quantitative traits locus. So as you can see sa table, makikita nyo dyan yung ating mga species or yung mga species pala na may sex-specific markers. Meron din yung kanilang sex determination system, the identified techniques, yung kanilang marker type and quality, and the associated genes. So, andyan naka-list down lahat ng merong sex-specific markers or sex chromosome-specific markers na kasama yung kanilang mga associated genes. Andyan yung yellow catfish, Chanel catfish, African catfish, and so on. And the biotechnological approaches of sex control breeding in fish. Exogenous steroid hormone treatment could effectively induce sex reversal in fish. In consideration naman of food security, the feasible way is to use the hormone-induced sex reversal fish as parents to breed monosexual offsprings in aquaculture. Para makapag-produce or makabreed siya ng monosexual offsprings sa aquaculture industry. Base naman doon sa identification of X chromosomes specific and Y chromosomes specific markers naman, yung yellow catfish or olme number 1 is a novel aquatic variety, has been produced by using biotechnological approach for sex control breeding. The mass production of all females can be produced by the mating of XX males and females. So, mangyayari lang yun kapag nag-mate ang babae at lalaki para makapag-produce siya ng mga offspring, kailangan nilang mag-mate parehas. So as you can see, meron diyang methyl testosterone or the Y chromosomes, Y6 X chromosomes specific markers and the Y chromosomes specific markers. The XX and the XY the gynogenesis, yung blue is the male and yung blue is the male and pink is the female. Nakapag-produce siya ng parehas na lalaki at babae. So, yung kanilang mass production of all females. Pero mas nakakapag-produce sila ng all females. A schematic diagram of mass production of all females population through an integration approach of gynogenesis and the methyl testosterone of Y chromosomes markers and the X chromosomes markers. So, ayun lang and thank you.